Good evening, and thank you for joining us. I'm Dr. Ellen Kaplan, the CEO of UW Health. Welcome to our 2021 Wisconsin Medicine Livestream series. We will be bringing you programs one every other Wednesday evening now through June 2nd. Tonight, we focus on cardiovascular disease and how stem cell research and therapy are being used to address the disease that is the #1 cause of death across the globe. Our program this evening showcases a truly collaborative approach that integrates basic science research, clinical trials, patient care, and public education to not only treat heart disease in revolutionary ways, but also to learn more about how we are to prevent it. I'm pleased to introduce tonight's lineup of fantastic, remarkable scientists. First up, I'd like to introduce Dr. Tim Camp. Dr. Camp holds the Herman and Eileen Tushman Chair in Cardiology and is a professor of medicine in the Division of Cardiovascular Medicine and professor of cell and regenerative biology. He serves as director of the Stem Cell and Regenerative Medicine Center. Welcome, Dr. Camp.
Thank you, Dr. Kaplan, for that introduction. Today, I'd like to share with you some of our work on a big public health problem. Next. And that is heart disease. As Dr. Kaplan mentioned, heart disease is the #1 cause of death in the United States. It has been since about 1900. It's also the #1 cause of death worldwide. You can see in the graphic at the right that as we get older, the heart disease as a cause of death becomes more common and outpaces cancer. Heart failure also is a big problem. It's the most common discharge diagnosis from acute care hospitals. And all of this costs our economy and our healthcare system a great deal, estimated at about $350 billion in 2015. Next. And one of the reasons heart disease is such a problem is that the adult human heart doesn't respond very well to injury. And that's shown here in the photo of a heart slice after a heart attack or myocardial infarction. The blue area is scar tissue. That's where the heart muscle has died and is replaced by scar. And the remaining red brownish tissue is functioning heart muscle. But it's not hard to imagine that if you start losing more and more of that pumping heart muscle to scar tissue, the heart begins to fail or worse. So we're faced with a big problem here in cardiology. And as 25 years ago, as a newly minted cardiologist and physician scientist, I came to the University of Wisconsin hoping to address this problem, in part because there was such tremendous need. Next. And what I found is something that's shown a bit here. Now you might think I got my slides mixed up, but what I'm pointing out here is that many of the pivotal discoveries in stem cell research took place at the University of Wisconsin. And on this timeline, you can see the W's where there have been important discoveries here at UW as well as around the world. But one of the fortunate things for me that happened is I met someone named Jamie Thompson in the mid-1990s who told me he could get human heart cells in a dish. And I thought, well, that's interesting, but I was a skeptical scientist, but it turns out he was right. And he had found embryonic stem cells that could generate heart cells in a dish, which I realized, and many people realized, is an incredible source of cells for research and potentially therapies. So that introduced me to a whole new area of thinking and research that we've followed over the last 25 years. Next. So let me just tell you a little bit about these special human pluripotent stem cells. The first type that was described and discovered are embryonic stem cells. They're derived from surplus in vitro fertilization embryos. In other words, embryos left over from fertility clinics that are donated to research. And then the cells are removed. And what Jamie discovered was how to get them to grow in culture. And they can self-renew indefinitely. That means they can keep growing and maintain their same properties. We don't know how long, indefinitely. We have some of the lines that Jamie derived in the mid-1990s still growing in our lab today, as does he and many others around the world, actually. The other special property about these cells is that they can differentiate or form any cell type in the body, including cardiomyocytes, which is important for our discussion today. The second major type of human pluripotent stem cell are induced pluripotent stem cells. Now, these are cells that started out as blood cells or skin cells, but are reprogrammed using factors that we learned about from the embryonic stem cells to make them like embryonic stem cells, so they're called human-induced pluripotent stem cells. Another major discovery by the Thompson lab and also by Shinya Yamanaka and colleagues in Japan. So these are special cells that have unique properties that are very useful for us, and I'll show you why. Next. So you may say, well, you can grow these cells and have them in a lab, in a dish, so what good are they? And to me, they're so important if you're interested in human health because they're a source of human cells for research studies. And many human cells, as I've suggested, are difficult to get our hands on. You can get a blood sample or a skin biopsy, but heart muscle or brain, that's hard to come by for obvious reasons. These also provide cells that can be used, for example, for drug discovery and drug safety testing. So you can think about things because you can get a genetic diversity of cell lines like clinical trials in a dish. So saving patients the risk of testing new drugs and doing it in the laboratory. It can also provide cells that can reflect genetic diseases. And so we can have models of human disease in a dish to better understand them and develop new therapies. And I just show you in the graphic at the right here why it is so important for some diseases to use human cells compared to our workhorse, which is the mouse that's been used for so much biomedical research. The upper slides, upper traces show you the electrical signals or action potentials from a human heart cell compared to a mouse heart cell. And you don't have to be an electrophysiologist to notice they're quite different. And those differences are due to many different changes in the ion channels and proteins responsible for that electrical signal. So human cells are better to study human disease. Next slide. And so one of the requirements to allow this research to really move forward is that we had to find better ways to make cardiomyocytes in a dish. And so this just schematically shows you a couple of protocols or recipes we came up with to make heart muscle cells in a dish. And we get whole contracting dishes of heart muscle cells where we can have tens to hundreds of millions of cells generated in an average lab every week if we want to. So it's something that's accessible now to many labs around the world. Next slide. And we get different types of heart cells. The schematic on the left shows you one way to tell what the different heart cells are in the heart. From the upper part of the figure where the pacemaker is, you see a bullet-shaped electrical signal there, down to the working myocardium where the action potential or electrical signal is much broader and takes a longer time. And if you look to the right in the slide, you can see the action potentials or electrical signatures that we get from different cells in our dish look like some of the cells that we saw in the native heart. So that's good news. We can get appropriate heart cell types in our dish to study. Next. And so one of the most exciting things about doing stem cell research here at the University of Wisconsin is we get to work with fantastic trainees that are the future of the field, and we get incredibly bright MD, PhD students like Mitch Berman, who was in my lab, who is a creative thinker. And one of the challenges that we faced with our iPS or ES-derived cardiomyocytes is that when they first form, they're very immature. They're like baby cardiomyocytes. But if you want to study adult heart disease, you've got to mature them. And the cells change dramatically during development. So Mitch discovered, really completely unexpected to most scientists, thinking a way to use a compound called poly-IC that dramatically accelerated the rate that the cells, so to speak, grow up and become more mature cardiomyocytes. So an important discovery that Mitch made, and exciting to be involved in the training of such great students. Next slide. What many of us are really looking forward to, and you'll hear from Dr. Raval, is cardio-regenerative medicine, because as I mentioned, many of the most common forms of heart disease are associated with irreversible heart muscle loss and also congenital heart disease. We may need to build up certain areas of the heart that didn't form. So the schematic just shows one idea where you can take a patient's cells, blood cells, for example, reprogram them to iPS cells, and then allow them to differentiate into the different cell types that can be used to repair the heart muscle. And you'll hear more about that from Dr. Raval. Next. Finally, I'd like to highlight one of the other features we have here at the University of Wisconsin, and that's the Stem Cell and Regenerative Medicine Center that is designed to foster these advances by getting collaborators from multiple disciplines across 30 different departments on campus, 5 different colleges, from UW Health, the School of Medicine and Public Health, includes chemists, statisticians, engineers, a remarkable collection of investigators interested in stem cell research working together to really bring the promise forward. We do things such as host meetings, next, and also importantly, we fund pilot grants. And pilot grants are opportunities to try higher risk ideas that may lead to remarkable breakthroughs, such as Jamie Thompson's discovery of human embryonic stem cells. Those are programs that aren't typically funded by federal agencies, so we rely on philanthropic support for those. We also provide training opportunities for students and outreach. So I'll thank you for your attention and look forward to questions.
Great. Thank you, Dr. Camp. You did a great job of taking a very complex area of science and making it simple in a way that all of us can understand. Now I'd like to introduce Dr. Lee Eckhart. Dr. Lee Eckhart is the Gary and Marie Weiner Professor of Cardiovascular Research and a faculty member in the Division of Cardiovascular Medicine within the Department of Medicine. She serves as co-director of the Inherited Arrhythmia Clinic. Welcome, Dr. Eckhart.
Thank you so much for the kind introduction. I am honored this evening to speak with you about a project that we're developing that's going to use some of the same technologies that Dr. Camp just spoke with you about to try to understand more of the mechanisms as well as develop ways of treating individuals who have suffered an unexplained type of arrhythmic sudden death. Next slide, please. Before we necessarily talk about arrhythmias, I'll just remind you about what a normal heart rhythm is meant to do. Your heart, shown here, is really an electrically driven pump. It goes about 60 to 80 times a minute. That's 100,000 beats a day, 35 million a year, and about 3 billion heartbeats over a lifespan. Next slide. If the rhythm is disrupted for more than a few seconds, this can result in loss of consciousness. And after a few minutes, irreversible multi-organ damage and death can occur, as shown on this slide from a patient who has several normal beats and then followed by chaotic arrhythmia. This is known as ventricular fibrillation. Next slide. Sudden arrhythmic death is not uncommon. In fact, it's very common. Over 300,000 individuals in the United States suffer a sudden cardiac arrest every year. Next slide. A number of these individuals will have had heart attacks, such as Dr. Camp just explained, or have structural heart disease. But those without structural heart disease can sometimes be diagnosed with certain types of inherited arrhythmias and that fall into certain phenotypic characterization categories. We can then understand their genetics, and then in the research lab, we can use our methodology of science to be able to understand those arrhythmia mechanisms even further, and then the idea is that this can then relate back to patient care. I can tell you, though, that not everyone, next slide, as an example of this, a patient of mine's rhythm strips are shown on the top. You can see that some of the rhythms are fairly chaotic, and she has these chaotic rhythms whenever she's under any type of stress, and that can be emotional or exercise-related stress. We did phenotyping for her as well as clinical genotyping and found a specific gene mutation, engineered that gene mutation into a mouse model, and then as it turns out, the mouse has the similar type of arrhythmia under stress as the patient does. So we can use that mouse model then to both understand the mechanism of arrhythmia as well as to develop treatments for this specific patient and then that specific syndrome. Unfortunately, not everyone fits into that neat little package of a specific gene mutation that goes with a specific syndrome for which we can base treatment recommendations on. Next slide. I'll give you another example. A 25-year-old woman who suffered a cardiac arrest while just riding in the car with her boyfriend. She said, I'm going to pass out. And then she did. He saw that she was in trouble, called the paramedics, and then did CPR. The tracing shown underneath the cartoon picture is her rhythm tracing when the paramedics arrived, which shows ventricular fibrillation. And then she's then shocked back into normal rhythm. Next slide. We did deep phenotyping for her as well as comprehensive genotyping. And despite the fact that, as you can see by the traces on the top left, as well as the two graphs below that, she has a lot of abnormal electrical firing. Each of those black spikes represents either the percentage or the number of beats that she has that are abnormal per day. Unfortunately, she didn't necessarily fall into a specific type of arrhythmia syndrome. She had no biomarkers that suggested one of these syndromes, and her genotyping only showed a variant of unknown significance. This is really problematic because not only do I not have a syndrome necessarily to be able to base her treatment recommendations on, I don't have a genotype for which we can do cascade screening for her family to assure her all of her many brothers and sisters, nieces and nephews, distant cousins, and even her own children down the line, that they will not suffer the same fate that she does. Next slide. So with more questions necessarily here than we had answers, it's really difficult to be able to prognosticate for this patient. It's difficult to then give her treatment recommendations for how are we going to necessarily address your arrhythmia to keep you from having the same episode that you had when you were in the car. Next slide. This is actually not that uncommon. This patient example I'm giving you happens about half of the time for those individuals who have a primary electrical sudden cardiac arrest. And this is in databases around the world, including some of the largest databases, for example, this one out of Canada. And even more difficult is that many of these individuals also harbor genetic mutations that are of uncertain significance, meaning they're not quite normal, but we're not really sure if they're pathogenic. And so we can't do anything with respect to cascade screening for the rest of the family. Next slide. If you try to take those people's genotype and phenotype and stick it into a specific syndrome, then it's like putting a square peg into a round hole. The patients don't respond to those treatments necessarily, and the management for those individuals is chaotic. Next slide. And I think the root of the problem really is that the clinical tools we have to decipher a treatment method are inadequate, and treatments are based on well-intentioned and sometimes and oftentimes ineffective guesswork. Next slide. I see this as both a problem, but also a giant opportunity that we have to harness the power of the and technological developments that we've developed, not only for creating iPS cardiomyocytes, as Dr. Camp described, but then also some of the platforms that we've been able to engineer to be able to create a model of a heart in a dish. So my project really is that we're going to take induced pluripotent stem cells, or we'll take blood from patients, make induced pluripotent stem cells, drive them to cardiomyocytes, and then do high-level arrhythmia phenotyping of those patients. We'll also engage in proteomic investigation, as well as a deep genetic study. Next slide. With our engineering colleagues in Dr. Wendy Krohn's lab and her graduate student, Brett Nabwaki, shown here, we've been able to design certain platforms for the cells to grow on so that they can develop cell-to-cell connectivity, as well as show size and shape that mimic a normal heart cell, so that you really can have more than just a typical cell culture of cells beating in a dish, but cells that really look like heart cells. Next slide. And the cartoon sort of pinkish red and black picture on the upper left, you can see a patient of mine who we've made iPS cardiomyocytes from, and then a student in my lab, Monica Calori, then is studying those. And you can see that some of the traces, the colored traces there look pretty normal and regular, and some are very fast and chaotic. So this patient's cells have certain arrhythmias. Then the idea is that we'll then use the cells in the dish and apply antiarrhythmic drugs on top of those to be able to design better treatment methods for these patients. Next slide. So this is really an opportunity. Instead of trying to put square pegs into round holes, we'll do deep genotyping, as well as try to understand the functional abnormalities that some of these variants of unknown significance are, arrhythmia, characterization, and then find medications that are already FDA approved that we can repurpose for these patients to try to prevent arrhythmias down the road. Next slide. I couldn't do any of this work without my fabulous colleagues in the Cellular Molecular Arrhythmia Research Program and those in the UW Cardiovascular Research Center, as well as the Inherited Arrhythmia Clinic. Thank you so much for your attention.
Thank you, Dr. Eckhart. And I have to say, it's really interesting. This is a real great example of personalized medicine, both for the 50% that you can identify the genetics behind the arrhythmia, but also the way you're looking at those we can't identify today and creating a platform for discovery and saving more lives down the line. Our final researcher this evening is Dr. Amish Raval. Dr. Raval is an associate professor in the Department of Medicine and Biomedical Engineering and a member of the Cardiovascular Medicine faculty. He is the Director of Clinical Cardiovascular Research in the UW Options Clinic. Welcome, Dr. Raval.
Thank you, Dr. Kaplan, and thank you for the kind introduction. And it's my pleasure to be able to give you a short snapshot of the work that we're trying to advance in thinking about delivering cell-based therapies and regenerative clinical trials. Next slide. And so Dr. Camp initially alluded to the fact that heart disease is a very common problem. In particular, when a heart artery is constricted, it can lead to heart damage and a heart attack. That heart attack can thin the heart muscle and cause a scar. And on the right side is a cardiac MRI showing thin scar and a dilated ventricle. But it's not just about pictures, it's about people. And these people suffer, and they have shortness of breath all the time, often, and they often have many medications that they've tried, often are either poorly tolerated or don't work. Next slide. And so our lab is working on sort of a multi-prong effort to try to curb post-myocardial infarction heart failure. And we're taking sort of three different approaches. One is to look at ways of replacing scarred tissue. Another way is supporting the myocardium or the heart. And another way is to condition the cells that sort of are impacted by the infarct and the scar that may impart new blood vessel formation, modulate the inflammatory response, and potentially prevent programmed cell death. Next slide. So we'll touch on those. Now, in order to think about how we're going to get cells or scaffolds or conditioning cells into the heart, we have to have a way of doing that. And the approach that we've fallen upon is this approach where we take a catheter that is inserted through an artery, typically in the artery in the groin, although we could now do it through the arm, advance it through into the heart itself. And this catheter is deflectable and steerable and has a needle tip on it. And once that needle tip is engaged within the heart tissue into the heart wall, we can then inject the cells in a kind of a deposit, very similar to how a vaccine is injected into the muscle of the arm. On the right panel, you'll see labeled stem cells injected, and you can see the bright image plume of the cells located in the myocardium, and they stay persistently there, and we think that retention is important in order to improve function. Next slide. Now, this is an example of a clinical system that I've been using for many years, that has been used in the use of involved in clinical trials that use this device. clinical trials involving cells and protein delivery methods. The catheter on the left side is the MyoStar catheter, and it's got a straight needle tip associated with it, but the back end of the catheter is deflectable and steerable. And on the right side is sort of an electroanatomic mapping system that we are able to use to kind of a GPS coordinate system that can create a roadmap for us to know exactly where we need to inject the cells. Next slide. But there are deficiencies in that mapping system as it currently stands. As you can see, it's a fairly crude image, and that's supposed to be a heart. The black dots do represent injection sites, but it's a fairly low resolution image. The act of doing the mapping can be time consuming, and the electronics that are required to navigate the catheter itself and have it, bound to the global positioning system in there makes it stiff and a little bit cumbersome to use. So our lab early on decided, well, enough of that. Let's think about other approaches. And so we embarked and developed a MRI to X-ray fusion approach, where we took a pre-acquired MRI, nicely highlighted the area of the infarct, and then overlaid that onto X-ray. And by doing that, we can now see the infarct very clearly. By doing some more contouring, we could highlight valves or papillary muscles if we wish. So that we try to hit the areas that we want to hit with the cells and avoid the areas that we want to avoid. And this is we proved that this was more accurate, it was time efficient, and we were able to use less stiff catheters. Next slide. So this internal approach of taking a catheter, it's minimally invasive, it has a lot of appeal, and we're using it. And we're using it in this pig study, where we're injecting a combination of committed cardiac progenitor cells through induced pluripotent stem cell technologies developed by Tim Camp, combining it with a cell-derived matrix that's formed into a particulate developed in my lab to act as a binder for those cells. And we take the cells and the matrix and we inject it into the areas of the heart damage that we've created in the pig model. And we're looking to see if this combination either induces or suppresses arrhythmias, whether or not this improves viability and function using MRI, and of course, determining whether or not there are some favorable responses that could predict what we might see if we use this approach in human studies. So this is exciting and ongoing. Next slide. The cell retention technology that we're talking about starts to get formulated as a sheet. It's a cell-derived biomaterial that we can tune into an injectable and then inject in combination with cells. So on the right panel, you'll see yellow labeled cells combined with the injectable matrix that stay persistent along the needle track, whereas blue labeled cells that were without the matrix are seen quickly escaping through the native vasculature. Next slide. So that's the approach that we're taking for looking at combinations of cells and scaffolds. But what about heart conditioning approaches? Well, this is a clinical trial that we're involved with. As we've come to know, the bone marrow harnesses a number of interesting cells that, under the right conditions, could increase new blood vessel growth, modify local immune cells called macrophages, and prevent programmed cell death. The problem is that they don't spontaneously come out of the bone marrow in enough numbers to impart a therapeutic effect. And so we have to harvest them, aliquot them, and inject them back. Next slide. And so we're taking this approach in a very exciting human trial. It's a phase 3 human trial called the cardiac heart failure trial. And this has put University of Wisconsin a little bit on the map because we are the National Data Coordinating Center. I'm one of the national co-PIs, along with Dr. Carl Papine from the University of Florida. And we're taking patients very much like the patient that I had seen earlier with the prior myocardial infarction, who has symptoms of shortness of breath, who has tried all medication options but has persistent symptoms. Next slide. And this is an interesting study where we're taking the approach of injecting cells from the same patient back into that patient. So from that patient's bone marrow back into the heart, which does overcome some of the problems related to cell rejection that can occur in other methods. But to do this, we have to be sure that the patient themselves harnesses the types of cells that are of interest in sufficient quantity that are likely to work. And so to do this, what we do is a two-step bone marrow procedure where we do a one step that's a small volume of about 5 or 6 milliliters, like a tablespoon. And then if that patient's cells are determined to have the right mix, we then bring the patient back and then do another larger volume aspirate, process those cells, and then inject those cells back into the heart using a catheter approach like you had seen before. And the goals behind the trial is to see if this cell injection approach improves functional capacity in terms of walking distance, reduces symptoms, improves quality of life, and improves injection fraction. And of course, we want to reduce mortality and hospitalizations. And what's exciting about this study is that if it's proven as a phase 3 trial, then this could be potentially, and interestingly, could be the very first cell therapy in clinical practice in cardiology. Next slide. I learned long ago that it's very difficult to have the infrastructure to kind of see and process patients who have advanced heart and vascular disease without having a team. And so I have clinical research nurses who help me through the UW Health Options Clinic identify, screen, and consent, and then subsequently follow patients who are able to be enrolled into investigational cell, gene, and protein trials. Of course, along the way, we're also put on my physician cap and think about ways of optimizing them that maybe their own providers hadn't yet thought of, as well as take advantage of emerging technologies that are clinically approved, such as advanced therapies with newer ventricular assist devices and even cardiac transplant. Next slide. But with everything else, like everything else, there are opportunities and challenges. The opportunities exist. This is team science. I work with dozens of different groups around the university who help me and help our team come together, and we really try to advance this science. We have the right ecosystem and a strong track record of success because of those features. But research funding is always going to be a challenge. Fortunately, we've been fairly successful with federal, industry-sponsored, and philanthropic support. The gifts that people have offered have been very useful to us, even in small ways, in advancing our research. So that's really what I wanted to talk about, a small snapshot in the delivery aspects related to cell therapies and the clinical trial work that we're ongoing. And of course, be willing to answer any questions that anybody has. Thank you.
Thank you, Dr. Ravel. It's always fascinating to see how basic science research translates into clinical trials. And as a physician myself, I'm just always marveled by the innovation and the thinking that helps with that transition. Just amazing people with amazing creativity and talent. So thank you. We have received a number of insightful questions during tonight's presentations. I'd like to pose them to our panelists. Our first question this evening is for Dr. Camp, and I will read the question. 1 in 3 deaths is a result of heart disease, with the common diagnosis prior to death, heart failure. What are some of the factors that contribute to heart disease? Have there been any recent findings in underlying causes of heart disease that have been surprising?
Thank you, Dr. Kaplan. Certainly, we have some pretty well-known factors that contribute to heart disease. Risk factors, many of which are modifiable. That means we can fix them. Perhaps the worst actor is smoking. We all know cigarette smoking promotes heart disease. Other things like high blood pressure, diabetes, diet, elevated cholesterol numbers, sedentary lifestyle, all those things are well known to contribute to heart disease and appreciated by all cardiologists. Certainly, some families also have premature coronary artery disease as well. So I think as far as new discoveries and surprises, I'm going to bring it back to the stem cell world. And one of the surprises, the bad news about stem cells, is something I think that's been discovered in the last 5 to 10 years, is something called clonal hematopoiesis. And that means some blood stem cells change in a way as we age that makes them take over the bone marrow, and they have different behaviors that can promote atherosclerosis, that can promote the blockages in the blood vessels that causes heart attacks. So that was a surprising discovery that came actually through the stem cell world and is not such good news. So we have to figure out how to deal with that potential problem that is a problem of aging.
Great, thank you. And Dr. Eckhart, next question is for you. In unexplained sudden death, how does clarifying what gene is causing the issue help understand how to treat it?
Thank you. That's an excellent question. One of the reasons why it's helpful to understand what the genetic makeup is that the collection of various traits come together that may assist us in unraveling the mechanism of arrhythmia. And so there's probably an unexplained sudden death, unlike other sudden death syndromes like long QT syndrome or catecholaminergic polymorphic VT, where it's most likely a single gene mutation. We can really study that specific mutation deeply and then understand the arrhythmia mechanisms. For unexplained sudden death, if we understand what the battery of genes are that are pathogenic, then that may help us to be able to look at both contractile proteins as well as ion channels, and then see how those may interrelate and contribute to that specific arrhythmia syndrome. On top of that, it's also really helpful to be able to understand who else in that family may be at risk for the same fate as that patient. We're thinking now that it's probably more than just one gene. It's more likely to be what we call oligogenic, meaning the number of genes, and which is why you don't necessarily see the same type of inheritance patterns in these families as, for example, long QT syndrome or hypertrophic cardiomyopathy.
Great, thank you. And Dr. Raffel, I am curious as to the answer to this question myself. So at the last Wisconsin Medicine Livestream featured breakthroughs in biomarker imaging. How do you collaborate with the radiation oncology scientists to develop specialized heart imaging at UW?
Well, that's a great question. So in fact, there are collaborations that are ongoing and are proposed in the future with respect to using radio labeling specifically for cells and matrix tracking. So it's really important, and we're coming to a head where we have to know what's going on with these cells. You know, where do they reside? Where do they egress if they do? And really what happens, are there off-target effects in other organs? And so the fluorine-19 FDG labeling that's been done, fluorine-19 has been used for a long time, but it's been used now in our lab for labeling stem cells to see exactly where they are. The dilemma with any sort of radioisotope use of labeling is that it's about the half-life of the isotope and how long can you actually visualize it. And so, for example, fluorine-19 is something that we can monitor for a period of about 24 hours or so at most. And then it dissipates and then you won't really see it. So for that acute retention study, which is really where we've been doing most of our work lately, it's been a very useful tracker. But for things like scaffolds, it becomes very difficult. It's not easy. You can't just sort of attach it or mix it with fluorine isotopes. So there's we're using a carbon-related dating type system that we're proposing in a grant that hopefully we'll get, and we'll be able to use it to track what happens to these particulates from the scaffolds that we're injecting into the myocardium as well. So those are a couple of examples of how we're interfacing with the radiation oncology imaging scientists.
Great, it's a great example of cross-specialty innovation. So Dr. Camp, when we cut our skin, it heals. When we break a bone, it heals. But you mentioned that when heart muscle is injured, it doesn't repair itself very well. Why is that? Why is this different than other tissues in the body?
Well, that's more than $1 million question. I think there's a lot of us that would like to have that answer and are working on that question. The curiosity is that that description fits the adult human heart. It doesn't fit certain organisms like zebrafish. If you injure a zebrafish heart, it completely regenerates. if you take a newborn mouse and cut off the tip of the mouse heart, it completely regenerates. That doesn't happen in an adult mouse. We think there's more regenerative capacity in newborn humans as well. So something happens as we age that turns off the ability of the heart to repair itself. Many labs around the world are trying to compare neonatal tissues to adult tissues, different organisms, to try and figure out where the switches are and what the critical differences are. But somehow over evolution, we've lost the ability to repair our heart as adult humans. And I wish I knew the answer to that. I'll let you know when I do.
All right, thank you. I'll be waiting for the answer. Dr. Eckhart, your goal is to create a path to a more effective treatment for all of your patients. Do you have any remarkable examples of treatment you've been able to provide to your patients?
Thank you for the question. There are a lot of patients that come to mind. A lot of my patients are really remarkable. Running a sudden death clinic means that everyone has died before they usually make it into my clinic. So it's a wonderful group of people to work with. There's one individual that really stands out to me. I met him about a decade ago, and he was in his mid-60s at that time, very athletic, ate all the right foods, and otherwise had completely normal heart, but he continued to go into this rhythm of ventricular fibrillation. He was in the middle of an arrhythmic storm, and when I saw him, he had almost 100 shocks from his internal defibrillator in a period of 24 hours. So he asked me, just turn it off, because I can't live like this any longer. And he was seen by really good doctors. And I said, well, let me look at things a little bit. And I tried to think outside the box. We tried a number of different treatments. I ablated some of his premature ventricular contractions. We tried a different batch of medications. And now he's more than 10 years later. And we found a concoction of medications that really work. If he misses a single dose of his meds, he gets a shock from his device. And so for him, the concoction works. But I have to say that I feel like the story has a good end, but it's not because of evidence-based science. It's not because I had mechanistic insight into his arrhythmia syndrome. It's because we tried a lot of things and then we got lucky. And so my goal is really to try to not just be lucky, but to be really be able to provide better assurance to patients that this is the treatment that's going to work for you.
Great story. And you may be the first doctor that I've ever heard that said every one of my patients has died before other than a pathologist. So that is an amazing description of the work you do. Dr. Ravel, how are your patients referred to your clinical trials and your work with the UW Options program?
Well, thanks for that question. So the options programs, which has been around since about 2008, we've been involved with about 22 different regenerative medicine type studies, and have seen countless numbers of individuals. What we've learned along the way, we used to have kind of a direct access, patient right into the clinic access, but we found that a lot of patients, had illnesses that didn't quite fit the bill. Sadly, right now, we're still in early days of human trials. And so far in cardiology, we're mostly focusing on patients with kind of advanced heart vascular disease who have to have at least tried conventional therapeutic approaches first. And so typically speaking, the route of access is for a patient to seek help through their own provider, typically their cardiologist or surgeon, and that surgeon or cardiologist referring in through a referral process. However, if that patient does not have that kind of access, then they can simply call in to the UW cardiology clinics line, where a self-referral is possible, and I can give you that number. It's 608-263-1530. And that would then provide, and if they just ask for stem cells or some options clinic, or if they can remember that, then that would definitely get through to our nurses. I'll tell you though that patients should be patient, because many times they haven't yet been tried on conventional approaches, and that has to, of course, be tried first. Later on, as we get more advanced, as we get more sophisticated with the clinical trials, I think we'll be starting to open up into other areas that maybe people with more moderate disease as more of a prevention type strategy. But right now, we're only seeking people with very advanced disease.
Great, thank you. Dr. Camp, you worked with Dr. Jamie Thompson and showed us a great timeline of stem cell science. At what phase of stem cell science did heart disease become an important focus of treatment?
Thanks for that question, Dr. Kaplan. I think that's a little hard to answer in any definitive way. I would say that from the beginning of the discovery of human embryonic stem cells, investigators started thinking about the potential therapeutic applications for various diseases across organ systems. Perhaps too much so. That contributed to some of the hype and unrealistic expectations from some of the early stem cell research. But like all clinical therapies, it's a long road until you get effective therapies. So I think it was in sort of after induced pluripotent stem cells were discovered in the mid 1980s, 2000s, that we started to really get more and more serious about human pluripotent stem cells as a potential clinical entity, because the processing of the cells and the robustness of the cells had continued to improve. In addition, cells that weren't human pluripotent stem cells but were autologous, in other words, some of the cells that Dr. Raval discussed, had already started in clinical trials. So work by Dr. Raval and colleagues paved the way to make some of the clinical efforts at heart disease using human pluripotent stem cells possible, including the preclinical studies that he's doing right now in large animal models. And so it has been an idea since the stem cells were first described, but it's gradually being put into practice as we get better and better with manipulating the cells and delivering them safely. And also we have some hurdles to overcome, such as immune tolerance, just like solid organ transplantation. Immunosuppression is a big issue. That will be something we have to deal with many of our stem cell applications as well.
Great, thank you. Dr. Eckhart, a follow-up question for you on a genetic connection to heart disease. There are so many stories of families with a legacy of early death due to heart conditions. How recent is the discovery that there are genetic and hereditary connections to heart disease?
Thank you for the question. The recognition that there are genetic factors that can lead to heart conditions, severe heart conditions such as long QT syndrome, goes back hundreds of years. The first probably written descriptions in a scientific paper is maybe around in the mid 1800s, but then Javel and Lang-Nielsen and then Romano and Ward in the 1950s published a paper describing the genetic long QT syndrome. We didn't know the genes back then, but you understood the inheritance pattern for these individuals. So going back and using typical Mendelian genetics to describe an inheritance pattern, you could track that. Understanding the specific genetic abnormalities involved really was about the mid 1990s when the first long QT genes were discovered to be implicated in long QT syndrome.
Great, thank you. Our audience is very active tonight and the questions keep rolling in. This one's for Dr. Ravel. I have two questions for you. First, could you share some of the known reasons why standard treatments are not always tolerated by a patient? And second, could you tell us about the model of the heart we see on your chest?
Well, let me take.
You have a big heart.
Well, let me take the first thing first. So any therapy the most ideal therapy would target the disease of interest and exclude having effects anywhere else. That would be ideal. It's just utopian. It doesn't really exist. All therapies have the potential for inducing side effects, or worse, people could be resistant to them. So they're taking it and it may not be working very well. So those are a couple of things. For example, in the case of heart failure, there are side effects that patients could be have symptoms by, or have no symptoms. Symptoms could be lowering their blood pressure. That could lead to dizziness, lightheadedness, and fainting. Or there could be effects that don't cause any symptoms but are alarming, such as worsening of their kidney function or changing their other electrolytes and so forth. So these are all sort of off-target effects that we're hoping to avoid, but can happen in this very sick patient population. And then the second question is, this is a hard model. So like so I had done a research fellowship many years ago when I didn't have any gray hair, and at that time, most fellows often get a pen or a plaque or something. I got a heart. So I had finished a research fellowship in cardiology at the NIH, and this was my gift. And so what this is I'm going to lift it up here. I think you can see it. So this is a very large heart. It's not at all close to the size of a normal human heart. It's probably more like a cow. And you can see on the outside, it's got blood vessels, blue meaning veins, and red meaning arteries. And I could even open it up, and I can see valves on the inside and the myocardium. So you've seen some photographs or pictures, cartoons on our slides. But this was a model that, in a day when we had the ability to be together, we could, show it and bring it out and have people touch it and look at it more closely.
That's great. It's one of the nicest pieces of desk artwork I've seen, or a paperweight. So the last questions for all our panelists. It's been a great session, but for all our panelists, what makes you excited for the future of your work? And I'll start with Dr. Eckhart.
I think that we're at a point in time where a lot of the groundbreaking research in stem cell technology, as well as culturing techniques, and then determining what factors are needed to be present in those cells to make them a more mature cellular phenotype, that is really the foundation for which I can use that technology to build on, to really bring us to the next phase. Instead of just creating a model in a dish, I think we're at the cusp of then being able to use those cells, then to be able to impact what we can understand for arrhythmias and how we can employ different treatment mechanisms and methods for patients.
Great, thank you. And Dr. Camp?
So I think, to me, it's combining some of our recent breakthroughs. So stem cell biology, we've got a reasonable handle on how to grow and manipulate the cells. But the new breakthroughs in gene editing, CRISPR-Cas9, combining that with stem cells, for example, correcting a genetic mutation in a stem cell line that could then be used as a therapy, that's happening already in some disease areas. It's not in the heart yet, but that's certainly an exciting area that'll move forward. And also combining some of the breakthroughs in what are called omics technologies, ways to study at the cell level many different features in the cell, from its genetic regulation to the proteins that are expressed, are really going to advance our knowledge tremendously and allow us to use stem cells in even more powerful ways than we have been so far. So I think we're really on an exponential growth phase of a lot of this research right now. Exciting times.
Great, thank you. And Dr. Rao?
Yeah, so I think one of my things that I'm really excited about is the possibility of being able to do combinations of therapies, like we talked about, replacement, support, and conditioning, combine them together to create a more kind of a holistic therapeutic model for heart repair, and then being on the front end of this involving patients. And I think that is really where it's all pointing towards, and we're hoping to help people out with these types of treatments.
Great. Thank you. Great panelists. Just thank you for being with us tonight, and thank you for our audience for all joining us this evening and for your great questions. I hope you enjoyed tonight's program and that you'll continue to tune in for our final 2021 Wisconsin Medicine program on Wednesday, June 2nd, which is focused on antibiotic and antifungal resistance. You can sign up to receive a reminder email and watch past sessions at wiscmedicine.org. Thank you for your interest. On Wisconsin.